morning, everyone. No? How about now? Got it? Okay, let's get started. We're a couple of minutes late. We've got a very full agenda. Josette. Not a very formal introduction.
No point in being really formal. Hi, I'm Josette Lewis with the World Food Center here at UC Davis. We're really pleased to be able to host this important discussion here today. As a university, one of our core business is doing research, and an important aspect of that research is to inform science-based policy, an issue that, of course, we're here to talk about today. Another important avenue by which we contribute to society is through the development of translation of that research into technologies. And here at UC Davis, we have a lot of relationships across the seed industry in our state of California and globally. And that's also an important industry that's impacted by our discussion here today. And lastly, we're really pleased to have so many colleagues here from the federal government in Washington, D.C., because we sit here in California. California and Washington, our agriculture is called specialty crops, by and large. Yet we're the largest agricultural economy in the country and one of the top 10 in the world. We produce over half of the fruits and vegetables in the U.S. So we play a pretty important role in the U.S. diet and the quality of the U.S. diet. But equally importantly, we have one of the most diverse agricultural systems, not only in the types of crops that we produce, but also in the way in which we farm, the different types of farming practices and scales. And so this conversation here today is a really important opportunity for UC Davis to host you, and we're really pleased that so many people have turned out, and to host our federal agencies to participate in an important conversation that shapes all of these different aspects that are important to us as a research university. So I want to thank you again for the opportunity to host this, and I will be here all day and look forward to hearing the discussion. Thank you very much.
So is my lapel mic on? I'm a wanderer. That's where I'm most comfortable, so I'm going to wander around a little bit. I'm Mike Furco. I'm with the U.S. Department of Agriculture. The agency I work with is the Animal and Plant Health Inspection Service, and I'm APHIS Deputy Administrator for Biotechnology Regulatory Services. So it's my shop in APHIS that has the piece of biotechnology regulation for USDA. I really like to thank everybody. This is a great turnout. It's more than we were expecting, which is great. I'm looking at the list of folks who are registered. It's extremely diverse. We have folks from a broad range of groups, interest groups. That's great. We should have a lot of interesting discussion today. One of the core themes of this effort that Robbie Barbero with the Executive Office of the President and others in the Executive Office of the President are working towards is better communication in this space. So I'm asking my colleagues today to think about communication. We tend to use jargon. We use not only scientific jargon sometimes, we use a lot of regulatory jargon. And as regulatory agencies, we have to be very aware of how we're using the authority that's been granted to us by Congress through legislation and then through regulations that we write. And we have to stay true to the authority that has been granted to us. We can only exercise authority that has been granted to us by Congress and that we ourselves have written into regulations. So if you hear stuff from us that seems really weird, go ahead and ask the question. You know, why are you saying it that way? There's going to be multiple opportunities today to talk about those things in the breakout groups, various question and answer sessions, and I look forward to the opportunity to help explain why we use such strange language sometimes. So looking at the agenda, we are doing some tweaking on the agenda because, as I said, this is a larger group than we anticipated. Primarily, it's a larger group in-house. It's much larger than either of the two previous meetings in White Oak in Maryland, and also larger than the meeting that we had in Dallas. So we'll start off with our our next speaker will be Robbie Barbero from the White House Office of Science and Technology Policy. And then we'll have the speakers you see at the table. We'll be talking about the work that we do in this regulatory space, the case studies, 3 case studies in particular, then a short break, and then the breakout sessions. And with the breakout sessions, that's primarily where we're going to have a little bit of tweaking. Since we have many more people present than we expected, we're going to have 3 locations where the breakouts will happen. One of them will be in this room, most likely in this section. People are still registering and they're signing up for one of the 3 breakout sessions, so we'll see how those sizes work out. And then there are 2 breakout rooms down the hall past the registration desk. So it'll be in those 2 rooms and then probably in this section of this room. Now, at the end of that breakout listening session, we had intended to have folks from each of those 3 groups come back here and do a verbal report out. We'd like to do other things. We'd like to leave time at the end for all of the public comments. So what we've decided to do is at the end of the breakout listening sessions, we'll go right into the public comment period, and then the rapporteurs and the facilitators of those 3 breakout groups will work together over the next few weeks or so, and then all of the information from those breakout listening groups will be posted on our websites. They'll be made public in a variety of different ways on the regs.gov area where this meeting has been located. So you won't have that verbal report out today, but there will be a written report out made available in the near future. Also there will be a transcript of the proceedings.
Can you explain, are the public comments going to be recorded in any way for people to feedback to?
Are they going to be recorded?
Recorded or submitted in one way?
They will be recorded, absolutely. The entire proceedings are being recorded.
Will they be posted, or will the public comments be posted in one way or another?
I believe they will all be part.
The whole meeting's being transcribed.
The whole meeting's being transcribed, so everything will be available. And in my experience in the previous 2 meetings, you can submit written comments as well. I mean, with so many people, at this point, we look like we have about 50 people registered to make public comments. You know, we were thinking 3 minutes each, and if we give everybody 3 minutes, they're going to be kicking us out of this room. Our contract goes until 3 o'clock. So we're going to give everybody a chance, and everybody who is registered to make a comment will get a chance. But what that means is that we might have to be tough sometimes. You can't see it here, but when you come up to make your public comment, there's about a 30-inch LCD screen right here. It counts down from 3 minutes. And we're going to have to stick to that because my primary value here today with the public comments is give everybody a chance to speak who has registered to speak. So I'm sorry if that means somebody gets cut a little short, but you have an opportunity to submit any comments you want in writing. Any quick questions before I turn it over to Robbie? Thank you.
Great, thank you very much, Mike. Thank you, everybody, for being here. I would like to note that just as in a classroom full of students, the first row is almost totally empty. So even adults prefer not to sit in the front row. Just an interesting note on the sociology of people here. So my name is Robbie Barbero. I work in the White House Office of Science and Technology Policy. And I'm going to provide a brief background on what we are doing and why we are doing it, and help you understand the process and where we are in this process. So in 1986, the White House Office of Science and Technology Policy issued a policy document that described the relative roles and responsibilities of the 3 primary agencies that had oversight over the products of biotechnology. That document was called the Coordinated Framework for the Regulation of Biotechnology. It did not endow new authorities onto those agencies, but what it did was it described how they would use their existing authorities in order to evaluate the products of biotechnology. In 1992, that document was updated, and then under this administration, and then the coordinated framework has not been updated since then. And so a few years ago, this administration issued an executive order that was not focused on biotechnology called Improving, oh, and you know what, do we have the, can I have that clicker so I can get you to the right? So Mike left us behind when he was wandering. All right, here we are. So this administration issued an executive order describing how all regulatory agencies could improve regulation and regulatory review. And then last summer, in July, the Executive Office of the President, which includes the Office of Science and Technology Policy and some of our other policy councils in the White House, issued a memorandum drawing on the principles articulated in those 3 documents that are on this list ahead of it, essentially directing the EPA, the FDA, and the USDA, which are the 3 agencies that are sitting here at this table, to modernize the regulatory system for biotechnology products. And there are 3 primary tasks in that memorandum, and I will distill it down here for you. And that memorandum is available on the website and in the docket, and so you can access it there. Those 3 tasks are, first, to update the coordinated framework to clarify the current roles and responsibilities of the regulatory agencies. So that will mean that these agencies will say, here are the products over which each of them have regulatory oversight. The second was to commission an expert independent analysis of the future landscape of biotechnology products. And then the third was to issue a strategic plan that these 3 agencies would follow in order to make sure that the regulatory system for biotechnology products is well prepared for future biotechnology products. This memorandum further identified both the goals and laid out some guidance for those agencies to follow. And I will loosely read here off of this. So that the essential guidance was that agencies should ensure that biotechnology products, they regulate biotechnology products, and they should continually strive to improve predictability, increase efficiency, and reduce uncertainty in their regulatory processes and requirements, and that these improvements should maintain high standards based on the best available science, and that deliver appropriate health and environmental protection, establish transparent, coordinated, predictable, and efficient regulatory practices across these agencies with overlapping jurisdiction. And part of what you will hear from us today when we discuss the case studies is how this coordination actually happens for actual products. And then to promote public confidence in the oversight of products of biotechnology through clear and transparent public engagement. And this is to follow up on Mike's point, especially when we were discussing these case studies. You will have an opportunity, once we walk through each of the case studies, to ask questions for clarifying questions. We may not be able to get to all of those questions, but we'll certainly do our best to try to answer them. Now, these are principles that guide these regulatory agencies to this day that are drawn from the 1986 Coordinated Framework and from the 1992 update. And in the interest of time, I will not go through all of these, but these slides will be made available to you on the website and in the docket on the back end. I think that a few things here that are important to note is that this system is really intended to be based on science, and it is a risk-based approach. And so you will hear these agencies talk about these tenets as they're describing the authorities that they use and how they use them to ensure the safety of the products of biotechnology. There's a real strong emphasis also on coordination and working together whenever there are abutting or overlapping authorities. Okay, so where are we now in this process? Well, in July, as I said, the memorandum was issued. And shortly after that, we did what every good bureaucracy does, that we formed a working group. So this working group is called the Biotechnology Working Group. It's formed under an Emerging Technologies Interagency Policy Committee. And this working group has representatives from the Executive Office of the President and each of the 3 agencies, EPA, FDA, and USDA. In October, this working group posted a request for information on the Federal Register with some description on what we were doing and why we were doing it, as well as some questions that were posed in order to help identify information that would be useful to accomplish the 3 goals that were laid out in that memorandum. There were over 900 comments received on that, and it was a very useful forum for gathering information. During that open comment period, the first of the public meetings on this memorandum was held at FDA, and we had over 300 people either watching on webcast or there in person, and received public comment at those. The transcript from that meeting and all of the relevant materials are available in the docket associated with this as well. Make sure that we're on the right slide. Okay. In January, the National Academies of Sciences announced that they were the entity that was going to be accomplishing the second of the tasks that was identified in that memorandum. So this is the landscape analysis of future biotechnology products. So the National Academies of Sciences announced that they were initiating this study. They are calling it the Future Biotechnology Products and Opportunities to Enhance Capabilities of the Biotechnology Regulatory Systems. And this study has 4 primary tasks. The first is to identify major advances and potential new types of biotech products over the next 5 to 10 years, to identify whether potential future products could pose different types of risk relative to existing products, to identify areas in which the risk or lack of risks associated with biotechnology products are well understood, and then, to the extent that's possible, identify what scientific capabilities, tools, and expertise may be useful to the regulatory agencies to support oversight of these potential future products of biotechnology. The academies have a website up for this now. Just 2 weeks ago, the academies announced the Provisional Study Committee. And by provisional, this means that they are taking public comment in order to identify potential conflicts of interest for this committee. So you can go to this website and look at the committee and submit a comment on the committee. And they also announced that the first public meeting on this study will be happening on April 18th at the National Academies building in Washington, D.C. Now, back to the work that this interagency working group has been doing. So since November, the interagency working group has reviewed all of the public comments that were received in response to the request for information. And using that information along with information gathered at the public meetings is in the process of both drafting this update to the coordinated framework and the long-term strategy. The second public meeting was held at EPA on March 9th. And then today we are here for the third of the 3 public meetings. And then this spring and summer timeline, the draft to the update to the coordinated framework will be released, and so will the long-term strategy. And it's important to note that that draft to the update to the coordinated framework will be placed into the Federal Register, and there will be an opportunity for the public to comment on it prior to it being finalized. At that time, all of the materials that are associated with these 3 public meetings will be in there as well, and so if you are on the webcast or are here today and would like to submit public comment in response to these meetings, when the docket is opened and the update to the coordinated framework is placed in there, that will be the time when you'll have the opportunity to place comments in the Federal Register. Okay, with that, let's get into some of the details now. So as Mike said, first we're going to walk through some case studies of hypothetical products. And these are products that, these are case studies that were written by these agencies, and they are really intended to give you, to illustrate the roles and responsibilities of these regulatory agencies vis-a-vis various products of biotechnology. Before we start into those case studies, we thought that it would be helpful to give you a very high-level snapshot of the authorities that each of these agencies use and the protection goals that are associated with those authorities. So when I say authority, that also could mean statute. So in this table here, you'll see that there are 3 agencies that are listed, and I believe 6 statutes that those agencies use. in order to oversee products of biotechnology. We'll give you a very high-level overview of those, what each of the protection goals associated with those are, and then start to talk through the case studies. It's worth noting also that in addition to this table, there is a table that is in the that's in the docket and that is available on the website. That is a much longer table of authorities and oversight that you're welcome to use. That information will be incorporated into the update to the coordinated framework when it's released, but that's another place where you can get a little bit finer grained detail on this. All right, with that, let's go ahead and review some of these authorities. Is this going to work? We'll see if that microphone works. Yeah.
Is this on? Okay. I'm Neil Hoffman. I'm with the USDA, the Animal Plant Health Inspection Service. We have two programs that regulate biotechnology. One is Veterinary Services, and they regulate under the authority of the Animal Health Protection Act. And this shows up here their protection goal is to protect livestock from animal pests and diseases. And on the plant side, we have Biotechnology Regulatory Services, which uses the authority under the Plant Protection Act to protect agricultural plants and agriculturally important natural resources from damage caused by organisms that pose plant pest or noxious weed risks. And I will turn this now over to my colleague from the EPA, Mike Mendelson.
Good morning, this is Mike Mendelson, and I'm going to talk about the pesticide part of EPA. My colleague Mark Siegel will talk about TSCA. Under the Federal Insecticide, Fungicide, and Rodenticide Act, our protection goals are to eliminate unreasonable adverse effects upon man and the environment. And for environmental and occupational risks, this involves comparing economic, social, and environmental risks and benefits associated with pesticide use. For dietary or residential human health effects, the sole standard is the safety of exposure. And then in addition to FIFRA, which is the licensing statute, the pesticide registration statute, we have the Food, Drug, and Cosmetic Act, and under that act, the protection goal is to ensure dietary exposure to pesticide chemical residues in or on food are safe. I'll turn it over to Mark.
Hi, I'm Mark Siegel from the Office of Pollution and Prevention and Toxics at EPA. And the statute that we work with is the Toxic Substances Control Act, as Mike referred to it, TSCA. Our protection goals, as indicated, deal with ensuring that all aspects of commercialization of chemical substances, manufacturing processes, distribution and commerce, use, disposal, et cetera, that with any combination of these activities does not present an unreasonable risk of injury to health or the environment. And I guess with that, I'll turn it over to Jason.
Good morning. Good morning. My name is Jason Dietz. I'm with the FDA Center for Food Safety and Applied Nutrition. FDA regulates approximately 20 to 25 cents of every consumer dollar spent. The statutes that we use to regulate those products include the Federal Food, Drug, and Cosmetic Act, as you've heard a little bit about from EPA, as well as the Public Health Service Act. And in terms of our protection goals for food, we certainly ensure that food, and that includes food for humans as well as food for animals, whether they be domestic companion animals or production animals, that food is safe, sanitary, and properly labeled. We also ensure that drugs for humans and animals are safe and effective, as well as biologics for humans. We also ensure the safety and effectiveness of medical devices for humans. We also regulate cosmetics. Many of you all may use. We must ensure that cosmetics are safe and lawful and properly labeled. And we also regulate tobacco products. Thank you.
Okay, great. Thank you, everybody. So go through a little bit of an overview of some of the data requirements that are associated with the authorities that we just talked about. After we go through this overview, then we'll walk through some of the specific case studies.
And will you be advancing the slides?
I'll do the slides for you, Neil. Yep.
So one thing we wanted to distinguish is between containment and confinement. APHIS regulates, and when APHIS regulates biotechnology, it's through a permitting type system. And we just wanted to clarify that we regulate confined field trials, we regulate movement, we regulate what is outside of enclosures, but we don't regulate inside an enclosure. So a containment system is not regulated by APHIS. The idea of by containment, we refer to things like greenhouses or growth chambers. The idea is to prevent exposure of those GE plants to the environment, and the probability of release should be near zero. And a confinement is like an outdoor field trial, and there we're trying to ensure that GE plants do not persist in the environment. Next slide, please. So to show a visual, this is a greenhouse with corn going in a greenhouse, and very often in the early stages, much of the research is done in contained facilities, such as a greenhouse like this or in growth chambers. And again, this would not be regulated. The next slide, please. If a GE organism was shipped from one facility to another, that movement would require some sort of an authorization. In contrast, confined field trials, the idea is that there are certain conditions imposed on those field trials to prevent the plant persistence. And some of these requirements that are imposed would be to, say, maintain an appropriate separation distance from nearby plants that are sexually compatible. And you can see here that there's some area that's fallow around the plant, so one can look for escapes. If you can hit, yeah. There are other requirements that have to do with cleaning equipment, so that plant material from that field trial is not left in planters, which are then moved off-site and might spread the organism. That there are restrictions on what can be grown in those fields the next growing season. There are restrictions on monitoring for volunteer plants, and there are restrictions to look for nearby sexually compatible plants. In the case where there are wild relatives, that those would need to be controlled, or that plant would not be allowed to flower, and that the GE seed from the test is not used for human or animal food. Next slide, please. So when one is doing a confined field trial, some of the considerations in terms of what kinds of conditions are used, you ask, is the crop outcrossing or self-pollinating? Is it weedy or invasive? Are there wild or invasive relatives? Can the plant or offspring persist after the test is over? And would the trial be expected to change the plant's weediness, would the traits be expected to change the plant's weediness, invasiveness, or reproductive biology? Again, these are all considerations about what kind of requirements should be used to help keep that field trial confined. Next slide, please. Then we have a process where a developer of a biotech crop or biotech organism can petition for non-regulated status. In that situation, if non-regulated status is granted, then permits are no longer required for the authorization for the movements, the interstate movement, importation, or environmental release. And in those kind of considerations, we look at the crop biology and the taxonomy. Are there genotypic differences between the GE crop and the non-GE crop? Are there phenotypic differences? We look at the field test reports and what data was collected during those reports and any other relevant experimental data publications and data upon which to base a determination. Next slide, please. There are two kinds of evaluations we do for a petition process for non-regulated status. One is, we call it a plant pest risk assessment. Again, when we talked about our protection goals are based on noxious weed or plant pest authority. And in fact, our regulations really are only based on that plant pest authority. We do a plant pest risk assessment to determine whether the GE organism poses a risk as a plant pest. And if not, we must deregulate it.
And Neil, can I interrupt you here just to clarify for folks here? So you just said your regulations, you said authority and regulations. So can you give a little explanation of what the difference is between those two?
So we use the Plant Pest Act has authority, and the Plant Pest Act has authority to look at both plant pests and noxious weeds. Our regulations were put together prior to the Plant Pest Act, which was cobbled together from a number of acts, and we've never codified the noxious weed provision of the Plant Pest Act in our regulations. And that is one of the things, you may know that APHIS is revising its regulations, and one of the objectives of revising the regulations is to consider whether to codify the noxious weed authority of the Plant Pest Act into the new biotech regulations, CFR 734.
So Congress writes the statutes and the agency writes the regulations.
That's correct.
Okay.
Sorry about that. I wasn't sure what you all needed to hear about that. The other evaluation that APHIS does at the time of considering a petition for non-regulated status is to do an environmental assessment or environmental impact statement. So we have NEPA obligations, and generally the NEPA obligation, if it's not a significant action, would be an environmental assessment, or if it was a significant one, and that's a term of art, but I won't go into, is to do environmental impact statement. And lastly, I'll just describe what are some of the components of a plant pest risk assessment. Again, our authority is to look at the plant pest potential, and so we consider whether the modification to this GE plant will create pest or disease problems for agriculture. We also consider whether it will become a weed, whether it will increase the weediness of sexually compatible plants, whether it will harm non-target organisms, example, beneficial or endangered species, whether it'll affect agricultural practices in a way which could create disease and pest problems, and whether it can transmit the genes to organisms with which it does not normally interbreed.
And I'd just like to highlight this slide. So that for those of you who might ask the question, what information would APHIS want from me in order to engage in a process? This is a good summary slide for you to look at. These are the kinds of questions or the type of information that are important to APHIS in order for APHIS to make a determination about whether there is a plant pest risk.
Thanks, Robbie. That's it.
Okay, Mark. So now we are switching to a different agency and a different statutory authority. And this slide is a parallel to the slide just previous to it. So Mark will talk about if you are developing a different type of product. So we're not talking about crops here. We're talking about microorganisms. The kind of information that is important to EPA as they use their authority granted to them under the Toxic Substance Control Act.
All right, so to start with, I have to make sure you understand that TSCA does not have explicit data requirements for submissions that receive pre-manufacturing oversight, which is what we'll be talking about. Rather, TSCA requires submitters to provide EPA with whatever data that are relevant to our risk assessments that they may have in their possession or that are reasonably ascertainable by them. So that's pretty broad. EPA, therefore, has provided guidance to assist submitters as well as to help ensure that EPA gets the data that it needs. The guidance is what we call our points to consider in the preparation of TSCA biotechnology submissions for microorganisms. So, okay, so I guess I was going to get to it, but I'll do it right now.
This is all background information. When we walk through the case studies, you'll get some of this explained to you. But this is background information that's sort of generic across many of the case studies, and we wanted to provide it to you. Recognize that it is nearly impossible to absorb all of this the first time through, but we'll do our best to sort of reiterate it and talk through it. Yeah. But that is a good question.
So on another slide, and later on they'll be explained, but a TERA, T-E-R-A, is a TSCA experimental release application. An MCAN, microbial commercial activity notice. So I will get to those exactly in just a bit. So anyway, this points to consider document is intended to provide assistance to those who come to us and make sure that, as I said, that we get the data that we need. So it's expected to be a broad presentation of the range of data categories that might be relevant to any risk assessment that we might encounter. Therefore, we don't expect every submitter to have to provide us with every piece of, or every category of information that is provided within our points to consider, since we have to deal with such a broad range of cases. We expect a submitter to go through the list of information that we provide and think about what's relevant to their particular case. That's points to consider. That's the consideration that we're expecting them to do. To go through this, look at the data types that are relevant, and we then provide within our points to consider guidance on how to present that data, how in some ways to collect the data so that we have it properly done. So what this slide does is illustrate the categories of information that are provided within this document. The very first thing we need to know is what is the product identity? What is the organism that you're working with? We need to understand how this organism may be related to other similar organisms for which we might be able to gather additional data besides that which is provided by the
And Mark, I'm just going to remind you we're already a little over time, so let's briefly run through this one.
So quickly, in the product identification, we need to know how the organism was put together. We need to know the obvious things, as everybody's talked about. Does it affect human health? Does it affect the environment? What are the explicit effects that might be considered? We are also concerned with worker exposure, so we look at releases from the worker environment and the use environment and exposure to the general population. For the TERA, the experimental release application, we're going to look at some specifics of the site, things like monitoring, issues that are unique to that set of experiments. Whereas for the MCAN, we look at the entire broad range of plausible uses of the microorganism beyond those that maybe the submitter may have considered they intend to use, because somebody else may want to use the same organism for a different purpose. And I think that's it.
And we recognize that this is a lot of information. There is much more detail available in the links that are in these slides. And certainly at any point you can also, reach out to these agencies and discuss the information. Yep. And in the case studies that have been handed out to you as well, some of these, this information is illustrated in a more practical manner. Okay, Mike.
Okay.
So now we are not talking about the Toxic Substances Control Act anymore. Now we're switching over to a different part of EPA.
Right. So in the Office of Pesticide Programs, we focus on the Federal Insecticide, Fungicide, and Rodenticide Act, or FIFRA, and FFDCA, or FD&C, the Federal Drug and Cosmetic Act. And so what these data that we require are used to do is to help us make the regulatory decision for licensing a pesticide, that there are no unreasonable adverse effects to man or the environment, and also the dietary risk assessment. Basically, there's a reasonable certainty of no harm from the residue of the pesticide in the food. And so those are the two assessments that are made, the regulatory assessments, and to support those, we require this data to perform risk assessments. So this is briefly for plant-incorporated protectants. We also look at genetically engineered microbes that have pesticidal properties, but I'm going to talk primarily today about the plant-incorporated protectants, or PIPs. So the first thing we look at is how to characterize the PIP, and
a plant-incorporated protectant is?
Thank you, Robbie. Okay, so the way we define a plant-incorporated protectant, it's the genetic material necessary for production of a pesticidal substance as well as a pesticidal substance in the plant. So, in short, it is not the plant, but it is a pesticidal substance and the genetic material necessary to produce that pesticidal substance in a plant. So, in assessing the risk for that, to make those two findings, we look at how we characterize the PIP by the transformation system, characterization of the DNA inserted into the plant. We look at inheritance and stability of the genetic insertion after transformation, and we look at the protein characterization expression of the pesticidal substance. Of course, for different types of PIPs, this will be tweaked somewhat. To look at the human health effects of PIPs, we mainly look at toxicity and allergenicity. We require an in vitro digestibility assay to look at allergenicity. We also require amino acid homology to look at both toxin and allergen possibilities. We look at heat stability, and we require an acute oral toxicity study. Again, these types of data are used to assess the human health risk. And lastly, I'd like to talk about how we assess environmental effects of PIPs. Again, we look at what's the pesticidal substances being produced in the plant. We look at environmental fate of the pesticidal substance, how long it persists in the environment, in the leaf tissue, in the soil, et cetera. We look at gene flow, how that genetic material can pass in the environment, and we look at non-target effects of the pesticidal substance. So this slide is basically an entire presentation normally, but it's a summary of the types of data we look at to assess the risk for plant-incorporated protectants. Thank you.
Okay, now we will hear from FDA about the types of data and information that are important to them when they are making assessments.
Thank you, Robbie. And this particular slide refers to foods from genetically engineered plants. When we talk about the genetically engineered animal in the case studies, we'll have a similar slide that details those specific aspects. I think it's important to understand that as a food producer, you have an obligation to ensure that the foods that you produce are safe and lawful. And to help firms ensure that they're meeting their legal obligation to produce safe and lawful foods, FDA has established a consultation process that allows firms to voluntarily engage with the agency prior to marketing to make sure that their foods, again, are safe and lawful. Some of the information that we typically consider, as you might expect, we want to know what the crop is, how it's typically used, what its intended change in the crop is. And in terms of what you might think of as science-based safety assessment components, we want to have a molecular characterization of the crop, what has been inserted into it, what is expressed from it. We want to then also know, are the expressed materials, are they safe? Is there any toxicity concerns, any allergenicity concerns? And we also want to look at the levels of key nutrients, anti-nutrients, and any endogenous toxicants that might be produced in the plant. And I think another important point about this is this assessment is performed on a case-by-case basis. And this is very important because it gives us the flexibility to ensure that the kinds of analyses performed are those most relevant to your crop. For example, the kinds of things that we might look at in corn may be very different from a tomato. And so one of the unique features of this program is that you have the ability to come to us very early in development, long before you have produced a product, to engage with us and talk to us about your product, and we can give you feedback on the kinds of safety and legal issues that you should be considering. And I think as we walk through the case studies, you'll hear that more than a few times. So with that, I would leave you with the message here that remember, your objective is to ultimately get to safety. There may be many paths that can get you there, but it is tremendously useful to come and talk to the agency prior to marketing, and the earlier in development, the better.
Okay. Thank you very much for those overviews. At this point, what we will do is walk through 2 case studies in the category we're calling products for human food and animal feed. I will present the case study. So you have these case studies in the packets that you received when you walked in. If you're watching online, they're available on the APHIS website. They're also available in the docket associated with these meetings. What I will do is give you a very high-level overview of them, and then each of the agencies will walk through why they would have oversight responsibility and why, and what the levels of engagement you would need to have with each of those agencies is. We'll do both of these 2 case studies in this category first, and then I will start a timer, and we'll take about 20 minutes of Q&A, and then when we run out of that time, we will stop and move on to the next category. So the first product here is case study number 1 in your packet. It's a hypothetical genetically engineered corn with pesticidal properties. It's a field crop used for food for humans and animals. It's engineered with a plant pest component to have pesticidal activity against certain insects. The product, corn, zeamase, is genetically engineered to express a protein with pesticidal activity. The gene encoding the protein is isolated from the bacterium Bacillus thuringiensis and is controlled by the cauliflower mosaic virus-derived 35S promoter. The construct is integrated into a binary vector and introduced into the corn genome using agrobacterium-mediated transformation. Also on that vector, and stably incorporated into the corn genome, is a gene that enables selection of transformants during R&D. So which agencies have oversight and why? Well, the USDA has oversight because the genetically engineered corn is engineered with a plant pest component. The EPA has oversight because the DNA codes for a pesticidal trait. And the FDA has oversight because the genetically engineered corn will be used for food for humans and or animals.
So the USDA would be involved in regulating this corn product. As I mentioned before, in the R&D phase, which is normally in a greenhouse or a growth chambers, that the only authorization that would be required from APHIS would be an authorization for interstate movement or import. Then when the developer gets to the point where they do small or large field trials, authorization would, in addition, be needed for the environmental release. We have a NEPA obligation. Our NEPA implementing regulations have a categorical exclusion from NEPA for confined field trials. The reasoning is that confined field trials are confined, so the exposure of that organism in the environment is negligible, and that there is not a need to do NEPA. And I'll be describing some examples where we have exceptions to that categorical exclusion. At the commercialization stage, usually developers petition the APHIS for non-regulated status prior to commercialization, and at that time we would, as I described before, do plant pest risk assessment and in complying with NEPA, we would typically do either environmental assessment or environmental impact statement. Are we going to take questions now?
Yeah, so can you hold that question? We can do that. We can hit on that one, but we'll, yeah.
Regulated and unregulated,
you want to briefly describe what you mean by
Okay,
regulated and non-regulated?
so if a G organism triggers our regulation, then it has regulated status. If at a certain time when information is collected about that organism, a developer can petition the USDA for granting non-regulated status. And at that time, they submit a dossier to the agency that includes some information. I had an earlier slide which described the type of information that the company might submit to us, and we would do a comprehensive review of the information they submitted and any other information in the literature that's relevant. And we would decide whether or not it poses a plant pest risk, and if not, if it does not pose a plant pest risk, then we would grant it non-regulated status, and permits would no longer be required for the importation, interstate movement, or environmental release of those organisms.
So I don't get exactly that. So you say we collect information. Who are you collecting the information?
The developer puts together a dossier.
Yeah, so just whoever designed the thing.
The person who's making the petition is a collection of information.
So let's we can take this one offline. I want to make sure that we have a chance to get through all of these. Yeah. Yeah.
So
I'll-
why don't I talk with you offline?
Okay, we'll do an interview after the.
Yeah, sure.
Okay. Neil, anything else on this, or you? Okay. But this is good. I appreciate these questions. I mean, I think that there is, as Mike indicated, a lot of jargon that gets used here, and this is important information.
Is that one of the major changes that we're doing with this meeting? Is it the deregulating and coming up with the standard?
So let's take this offline. I want to make sure that, okay, all right, thank you. Okay. Finished, Neil? All right, Mike.
Okay, I'd like to talk about what we look at with PIP. So that, again, the way EPA looks at it in the pesticide program is it's a genetic material necessary for the production and the pesticidal substance in the plant. So at small scale, largely EPA is not involved. That's under 10 acres of testing. At 10 acre, after larger than 10 acres, EPA requires an experimental use permit. So this is prior to the commercialization of the product, but at large-scale testing, we require the petitioner to go ahead and to obtain an experimental use permit, which is a permit that allows the field testing of the plant over 10 acres. In addition to that, in addition to the experimental use permit, EPA requires a temporary tolerance exemption. So the one is for the use of the material in the field. The other one is for the residue of the pesticidal substance in the food. So we require a temporary tolerance exemption for that. When we get to commercialization, EPA requires a permanent tolerance exemption, which is a regulation allowing the residue of that pesticide substance in food, as well as a registration. That's to register the plant incorporated protectant. The registration is a license that allows the sale and distribution of the pesticide.
Thank you. And for the FDA perspective, this is a crop that's going to be used for food, presumably food for humans and animals. So certainly that food must meet the standards in the Federal Food, Drug, and Cosmetic Act. Generally, for genetically engineered plants, we would strongly suggest that a developer complete a voluntary consultation with the agency prior to marketing, and that traditionally has been the process through which developers have entered the marketplace. While developers may not necessarily come to us during the research and development phase, as I mentioned earlier, the earlier you consult with us, generally the better for the agency and the better for you. It gives you a more predictable regulatory process, and decisions that you make in your research and development phase may impact the regulatory aspects of your product. So certainly as you move from research and development to small-scale field trials, And then to larger scale field trials, it's useful to consult with us because we can help give you feedback that you can use to incorporate into the design of your field trials and the information that you might collect as part of your field trials. And then once you have completed your trial work, you can then submit a summary of your safety and nutritional information to FDA. We have a team of scientists there that would evaluate it. Once we are satisfied that there are no outstanding food safety or legal issues, we will respond to you with a letter indicating that we have no questions about your safety assessment, and then we will post on the internet a memo that describes the consultation and a copy of the letter that has been sent to you. And I think, again, as I mentioned earlier, I think the key with this particular case study is the consultation well in advance of commercialization and early in development. That, again, I can't say it enough, but that makes it a predictable process for you and a predictable process for us. And it also helps us identify issues very early in the process. Thank you.
Okay, so now we will do the second case study in this product category. This is case study number 3 in the packets that you have with you. It is a hypothetical genetically engineered herbicide-tolerant canola. It's a field crop used as food for humans and or animals that is genetically engineered with a plant pest component to tolerate an already registered herbicide. This particular herbicide has not previously been used on plants used for food for animals. The product is a domesticated canola, Brassica napus, that is genetically engineered to tolerate an herbicide by increasing the expression of a gene found in the canola genome using the constitutive 35S cauliflower mosaic virus promoter. Extracted canola oils will be used for biodiesel production, and the remaining biomass will be processed into meal for food for animals, and the animal or products of the animal may subsequently be consumed by humans. The 35S cauliflower mosaic virus promoter and the canola gene are co-introduced into the plant using a biolistic approach. Because the canola gene confers resistance to an herbicide, no additional selectable marker is required. This particular herbicide, herbicide X, is already registered by the EPA, but is not yet approved for use on animal food crops. And in quotes there, new food use. So that's a term of art there. In this scenario, a single developer produces both the herbicide-resistant canola and the herbicide. So which agencies have oversight and why? The USDA has oversight because the herbicide-tolerant plant is genetically engineered with plant pest components. The EPA has oversight because it regulates the new use of the herbicide itself, not the genetic material used to engineer the plant. The FDA has oversight because the GE canola will be used for food for humans and or animals.
So the USDA would have pretty much the identical role for this product as the last one that I described. I'll just go very briefly. You would need authorization for movement during the R&D phase. You would need permits or notifications and authorization for the environmental release during the small or large-scale field testing. This particular product we would be very familiar with. It would be categorically excluded from NEPA during field testing. At the time of granting for petition for non-regulated status, then we would do plant pest risk assessment and do NEPA analysis would typically be an environmental assessment.
So as Robbie said, for EPA, we're not regulating the PIP. This is not a PIP. We're not regulating genetic material. We simply, in this case, regulate the chemical herbicide. So it's associated with the use of the plant. So in this particular case, prior to small-scale field trials, we don't have oversight. At the large-scale field trials, if a company were to do that, they would need to get an experimental use permit and a temporary tolerance exemption or temporary tolerance for the herbicide. In practice, most companies test at smaller scale. We don't really see EUPs for these that often. Prior to commercialization of the herbicide, this is commercializing the pesticide, the herbicide use for use on this crop. And why is this different than the current uses? Many times it's a different rate, a different timing of the application, or sometimes a different crop. And for those, we would need to get a tolerance for the herbicide to allow for the residues of the herbicide in the food. And we would amend the herbicide pesticide product registration or Another way that that's done is most companies would obtain a new registration for that use. So again, for EPA's side, we would look at getting a tolerance in place for the herbicide being used on that food crop, and also having a registration in place or an amendment to an existing registration that would allow the use of that herbicide on that crop.
And from the FDA perspective, again, this is a crop that we used for food, and so not much will have changed from the situation with corn, except that we're dealing with canola, and that is relevant to our case-by-case safety assessment, because our safety assessment will focus on the key aspects of canola in this instance instead of corn. And as I mentioned before, it's always good to contact us early in development and as often as necessary, and that helps produce a good product for you and a predictable regulatory process.
Okay, so we have now gone through these 2 case studies. What I'd like to do here is allow about 20 minutes for questions. What I'd like you to do is just raise your hand. We have some people with microphones. And so you've already had a turn. Let me do a circle around and get everybody else, and then we can come back. Basically, please say your name. And I think there's one here. Yep. And please make it a question. If it seems to me like it's not a question, I will just urge you to turn it into a question so that we can respond then.
Okay, my name is Belinda Martineau, and my questions apply to both of the case studies we've just gone over. They have to do with oversight. So you say that USDA comes into it because of plant pest components, but I thought that in other literature you've given out, you talk about whether it poses a plant pest risk. So components of a plant pest don't necessarily pose a plant risk. And then there are other GMOs that don't contain these plant pest components and wouldn't come under their regulatory oversight. And then with the FDA, of course, it's not really
so
oversight.
The question for USDA is?
The question is, doesn't the product have to pose a plant pest risk as opposed to just containing plant pest components?
Okay.
For USDA. And then for FDA, the oversight, obviously, a company could, since it's voluntary, not even show up for a consultation. And couldn't the FDA use its food additive authority to regulate a foreign protein added to a genetically engineered plant as a food additive, which was what it did for the first genetically engineered whole food that came through the FDA?
Okay, thank you. So let's start with USDA. The question clear to you, Neil?
Yes, so if a plant contains a plant pest component, it triggers our regulations, and then it needs a permit, needs some authorization in order for those 3 areas: importation, interstate movement, and environmental release. If it doesn't pose a plant pest risk, that plant pest risk is evaluated at the time of a petition for granting non-regulated status. So it is under regulation, it's requiring a permit because it has those plant pest components. If we determine it doesn't pose a plant pest risk, then we will grant non-regulated status and it no longer needs the permit. I don't know if that answered your question.
So you're saying that if there's a plant pest component that there's you feel that it's important at that point to assess for a plant pest risk, and that's why you do that?
Yes, we don't actually assess whether it poses a plant pest risk at the beginning of the process. And so if your question was whether or not, actually, I'm not absolutely certain about your question. You were making it, you're right, there's a distinction between a plant pest component and whether it poses a plant pest risk. If we determine it doesn't pose a plant pest risk at the time of our petition for granting non-regulated status, we will not continue to regulate it.
Okay. Jason?
Sure. Thank you. First, I think it's important that we recognize that all food must be safe regardless of how it's produced, whether it's produced through genetic engineering or some other process. The applicable provisions of the law apply. And one of the things that we do consider during the consultation process is whether the new protein is in fact a food additive that would require food additive approval. And if in fact we found that that particular protein or any other added substance would have to go through the food additive approval process before it could be lawfully marketed. And you are correct that in fact the kanamycin resistance protein in the Flavor Saver tomato was one case where a protein added to a genetically engineered plant was regulated through the food additive process.
Okay. Next question. You want to have one? Sure, you can have one more. Yep.
Okay, so you're with the White House, kind of. You're with the White House, right?
I work in the White House, yes.
Okay, right. So, are you familiar with Obama recently did, maybe 2 years ago, a behavioral science executive order? And if you are familiar with it, can you share with me, without me doing an ROI, in what manner is he using that executive order in this process?
So these questions are intended to be about the case studies that we have here in front of us, so that we can stick on the time here.
Oh, my bad. I'm sorry.
Yeah, no problem. That's all right. Why don't we find each other?
Is that something you could help me with later?
If we have time, let's find a chance to connect offline. Okay.
Okay.
Thank you. Over here.
Unless I'm mistaken, do any of these case studies involve cisgenic processes or any of the new gene drive processes? It seems like they all contain some kind of transgene component. Is that true? And if where do the cisgenic and the gene drive and gene editing procedures come under regulatory framework?
So, so these case studies and the discussion here is just limited to what's in these. To the extent that you have questions about other related issues, let's find a time to connect and talk about those. Are any of these cisgenic processes, or any of them gene drive processes, or are they all transgenes? That's the question. There are no gene drives in these case studies. Are there cisgenic? I don't believe there are.
And why was it chosen to not include the new technologies in any of the case studies?
So these case studies were derived in order to give an overview of how the current products
that are being developed or that are on the market?
To the best extent possible, to give an overview of the current roles and responsibilities for current products of biotechnology.
So can I assume maybe that there is no regulatory framework for the cisgenes and the new gene drive and SynBio technologies then?
So I don't think that's a safe assumption to make. Why don't we find a time to connect offline? There will be a lot of opportunity for this discussion during the breakout sessions.
I'd like it to be either in a breakout session or something that isn't offline.
So during the breakout sessions,
Thanks.
I would encourage you to bring this topic up and discuss it then. Okay? Thank you.
No one else has one. I have one right up this alley would be perfect with this. Okay, so.
Can you let's use the microphone so that we can get this transcribed. And can we get your name as well?
Yeah, I'm Josh Wilson. Hi, people. This is the first time I've ever engaged with any stakeholders, because all I do is write you guys letters, and I never hear anything back. So it's a big black hole, all my words. So this is kind of cool. It's interactive. Okay, so let's be hypothetical here. We take this product, and we deregulate it, APHIS. So now it's deregulated, so we can do whatever we want. It's open. We have no records. We don't need to ask the company to tell us how many hectares are going or anything like that. Doesn't matter. It's completely free to do whatever it does. And
so can you which of these case studies are you talking about,
So it's deregulated. We have this product, and now a seed seller wants to sell it to our public lands, our public land managers, and it's deregulated.
So which product are you talking about?
The hypothetical non-food product.
But which of the two that we just went through? So we talked about a, do you have the
Canola.
booklet in
That's-
front of you?
The canola would be fine.
Okay, so the genetically engineered canola that is
Right.
herbicide
Right, so
herbicide
this
tolerant.
thing,
Okay.
we found that this works great with our ARS programs and our public lands, and BLM is finding a place for it. Okay, so would the land manager, because it's deregulated, not have to be aware of that? So if a land manager is saying, well, I think this canola thing, I just see what it does and they tell me it does everything great, would there be any, because now that it's deregulated, would that bypass NEPA's act for the Bureau of Land Management to start applying this seed?
At one time, genetically modified crops that were deregulated were planted on parkland. The Department of Interior has changed their policy, and to my understanding, they are no longer using GE crops on parkland. As far as Bureau of Land Management, I imagine as part of the Department of Interior that would be.
It is under DOI, correct?
Yeah, so
is that
I'm not
different?
that familiar with the entire legal
can fill you
implications
in after.
of that policy, but
Yeah, I'll let
so
you
I
know.
think the answer is no, that they would not be using that. There are some cases, for example, in Colorado, there was some public space and there was interest in using that public space to grow GE sugar beets, and then there was opposition to that, and I don't believe that they are using that space. So in some cases, there have been public input about whether or not to use GE organisms on public space, and there's been some response to that public input when there has been an opposition to it.
And so, Neil, to the extent that someone from one of these agencies came to APHIS to ask about the status of a product, what would APHIS's role in that be?
Well, we would just say
deregulated.
if we have deregulated it, we have no longer have jurisdiction over it, and
But it is still patented. Does it have to be patented to be deregulated? I guess if somebody, well,
you know,
if somebody.
that's a different story. It doesn't have to be. There are some. We've already been doing this long enough that there are some in some crops that have already come off patent. So it doesn't have to be patented. No.
All right, great. So, Josh, let's move
Yep,
on to the
thank
next
you.
set of case studies. Thank you.
Thank you.
All right. So with our next set of case studies, and actually we're going to do one under the products for biomedical engineering. This is the hypothetical GE rabbit. It is case study number 8 in your packet. This is an animal that is genetically engineered to make a therapeutic protein, recombinant insulin, for treatment of humans lacking this protein activity. The product, the rabbit Ornithologous cuniculus genome, is genetically engineered to express recombinant human insulin for use downstream as a therapeutic protein in the treatment of human patients lacking adequate functional insulin. The human insulin coding sequence is controlled by a 5' bovine alpha S1 casein promoter sequence to allow expression of recombinant insulin protein in rabbit milk. The construct is microinjected into fertilized oocytes, and the issuing embryos are transferred to the oviduct of a recipient. Also encoded in the vector and stably incorporated into the rabbit genome are upstream and downstream regulatory sequences that enable expression of the included codon-optimized human insulin coding sequence and insulator sequences to minimize position effects at the locus of genome integration. Once a germline transgenic animal is identified as a founder animal, it is bred to establish a lineage of GE rabbits used in insulin expression in milk. Which agencies have oversight and why? The FDA has oversight because the recombinant DNA construct encoding the recombinant human insulin coding sequence is integrated into the genome of the GE rabbit. It is regulated as a new animal drug by the FDA Center for Veterinary Medicine, and the recombinant insulin purified from the GE rabbit milk is regulated as a human drug by the FDA Center for Drug and Evaluation Research. So it's important to note there that there are two components of FDA that work together in providing oversight for this product. And with that, I'll hand it over to Jason.
Thank you, Robbie. As Robbie said, this is an interesting case study because you have two FDA-regulated products, a new animal drug associated with the recombinant DNA construct in the rabbit, and the recombinant human insulin, which would be a new drug for humans. And there is a mechanism at the agency for centers to interact with each other for products just like this one. So first, once you have identified the founder animal and the lineage of animal that you want to evaluate for potential production, you would come to the agency and request that an investigational new animal drug file be opened, and you would do that at the Center for Veterinary Medicine. They're the folks who regulate all new animal drugs. And this investigational new animal drug file, or INAD, this would allow you to submit information to the agency as you collect it for investigational purposes only, because again, you're still in the development phase here. And then prior to clinical trials with the insulin, you would need to go to the Center for Drug Evaluation and Research and file an investigational new drug application there so that you could then begin your clinical trials using the insulin. And then prior to commercialization, you would need to submit a new animal drug application to the Center for Veterinary Medicine for the recombinant construct in the rabbit. So that would be your new animal drug application. Then you'd also need to submit a new drug application to the Center for Drug Evaluation and Research for the recombinant human insulin. And then both CVM would have to approve the new animal drug application, and CDER would have to approve the new drug application for your product to be fully authorized. And the standard that we would use is the standard that we use for all new animal drugs and drugs in general, which is that it must be safe and effective for its intended use. So if you want to go to the next slide. So we talked a little bit earlier today about some of the data requirements, and so this slide is intended to describe to you some of the kinds of information that would be included as part of an INAD, Investigational New Animal Drug File. In that particular case, you would include information about the species under study, the genes that you've introduced, what you would expect to be expressed, and in general, the phenotype that you are expecting. The INAD regulations do have some specificity to them in limiting what you can do, and you must keep the animals under containment at this phase. There is special labeling that would be associated with them because they are investigational new animal drugs. There would also be special record-keeping requirements. You'd also have to ensure the proper disposition of the animals, and you'd also have to notify the agency when you intend to move generative material from one location to another, and then some environmental considerations to consider with the National Environmental Policy Act, or NEPA, and many of them may help to be satisfied by the fact that these animals would be well contained, would not be entering the food or feed supply without some degree of authorization from CVM. In fact, to date, the agency has approved 2 animals that produce pharmaceutical substances, and neither of them are authorized to enter the food supply, and they are highly contained. So then to the new animal drug application, some of the information that you would consider there. Much of this is probably as you would expect. We want a product identification, molecular characterization of the DNA you intended to insert, and then a characterization of what you actually did insert, phenotypic and genotypic characterization of the animal. We'd also want to know about the phenotypic and genotypic stability of the animal and a plan that you have to ensure the phenotypic and genotypic stability over time. There would be food and feed safety assessment if the animal were intended to be used for food or feed. In this case, where we've had biofarm animals to date, they have not been intended to use for food or feed. And then you would also need an environmental safety assessment. That could include information such as your containment, the disposition of the animals, and that is a good example of a case where it's very useful to consult with the agency ahead of time so that you can receive their input and expertise in how to prepare your environmental assessment. And then you'd also need an effectiveness and claim validation. That is, does the new animal drug do what you say it does? And I will point out that we have a very good guidance on this. Guidance 187 lays out a number of these factors, and if you've not read it already, I would suggest that you take a look at it if you have more questions. Thank you.
Okay, so we'll do about 10 minutes of questions here. I see someone standing up here in the middle. And just for these various agency-specific documents that we're talking about, for example, if you wanted to find guidance 187, you could just do a Google search for GFI or guidance for industry 187.
Hi, thank you. My name is Dana Pearls. This is a great case study, especially given the approval of the GE salmon. So this example was a very clear example of something intended for therapeutic treatment for humans. However, as we've just seen with the GE salmon, this was the process that it went through as a new animal drug, despite the fact that there aren't medicinal therapeutic purposes for humans. And nor was there a full environmental impact statement conducted, despite a lot of controversial findings. So I'm curious, given the fact that the GE salmon and a number of other animals that are in the pipeline for approval don't serve therapeutic drug-like purposes, is this the same process moving forward intended for GE animals, and I don't know if GE insects would be there? How will these animals moving forward be regulated, and is it possible that there would be an independent committee, as there has been in the past? Would that be resurrected? And also, just to tag on that, a lot of these new GE animals being researched are using new gene editing techniques like CRISPR.
So Dana, I'm going to ask you to focus the question just on this case study to the extent that's possible. When you want to discuss some of these other issues, I think we should do those in the breakout sessions.
So I'd like for everybody to be able to hear, because this is a super specific example that doesn't seem to apply to a lot of the GE animals coming through the pipeline. So for those, is this the process that it will be ushered into, or is there a more robust process for things that aren't for human treatments?
So I think that these questions are best done if you can focus on a specific, do you have a specific case that you would like to ask about?
For example, Aqua Bounty has suggested that they will be putting another GE fish, the GE rainbow trout, on the market, or at least put it up for approval. Similarly to the GE salmon, this doesn't serve any medicinal purposes for humans, such as the case study. Will it go through this same process as a new animal drug, despite the fact that it is not a human or animal drug?
Okay, thank you. You got that, Jason?
So what I can focus on here today is the case study that's in front of us. And what I can explain is that in this particular case study, the new animal drug provision was used because the recombinant DNA construct in the animal fits the definition of a new animal drug because it affects the structure or function of the animal. And it's that structure or function clause of the new animal drug definition is what gives us the authority to regulate this as under the new animal drug provisions.
Yes, two questions over here. So we'll start with the woman in the front row. If you could say your name and your question, and then we'll go to the gentleman next in the next row.
Okay, Sally Fox. And my question is, what is in place to evaluate how the animal is affected by this insertion, the welfare of the animal?
Jason?
Great question. In fact, one of the aspects of this as a new animal drug, the agency looks to make sure that the substance is actually safe for the animal itself. It looks at the health of the animal.
And do you have, to the extent that you're able to talk about what some of those questions might be that you can draw? I'm assuming that GFI 187 provides some of those lines of questioning.
GFI 187 would be
do you have any of those you can elaborate on here, or do you think that's the best place to go?
I would say that's the best place to go.
So if you look at the FDA's guidance for industry 187, which is the document referred to here, you can find the types of information that the agency would have regarding how the health of the animal would be evaluated based on that.
The other point there, too, is remember that the safety assessment is safe and effective. And here what we're talking about safe is safe for the animal, and then of course if it were your production animal, it would be safe, would the food and any feed products from it be safe for humans, but in general you're saying, is it safe for the animal? So there's absolutely an animal safety component to this. And our folks at CVM take that very seriously.
Hi, good morning. I'm Arturo. I'm just curious as to how the FDA oversight would change if the recombinant protein expressed was, for example, like a cow casein for food products, as opposed to an insulin for as a drug.
So if it were a casein product instead of insulin, as I understand it.
So the question is if it's casein that's being produced instead of insulin? Okay.
Well, it may depend a bit also on the intended use of the casein. That would be an important factor. Is it intended for medicinal use? Is it intended for food use? We would take that into account. And you would still also have your new animal drug components. I think that's probably as much as I can elaborate on that without a lot more detail.
So this is an example of where the conversation would be with FDA, where the product developer would say, here is the exact the product details and the intended application.
And I think, Robbie, that's an important point. I mentioned that point quite a bit with plants, but the very same is true for genetically engineered animals. It is very, very important to contact the agency early in development because they can help make sure that you are meeting your legal obligations and help ensure that you are on a good track. So I would strongly encourage folks to reach out to the folks at the Center for Veterinary Medicine when they're interested in producing a new genetically engineered animal.
So did we have one in the back corner? And then we'll do over here.
So
name, please.
Oh, sorry. Pablo Ross, I'm a faculty at UC Davis. And so my question is a little bit of a tweak to the way that this animal is constructed. So instead of using a construct, let's say we take the very few differences between the rabbit insulin gene and the human insulin gene, there may be a few base pairs, maybe 10, 20, and we change those without using any recombinant product or material. Would that still be considered a drug, an animal drug?
I think I understand your question. However, what I would suggest is back to the point I just mentioned. That's an excellent kind of question to come to the agency and talk to them about and get the experts' views on your specific product.
Yes, Kenneth Oy from MIT. Now, the question that I have is really on the issue of safety and efficacy from the perspective of the rabbit. If we look at this example and view this in the context of other therapeutics, you would be expecting potentially issues to emerge over longer periods of time. If you look at the Japanese PMDA on regenerative medicines, or you look at the European Medicines Agency on therapeutics, they're experimenting with adaptive pathways precisely because it may take time for effects to materialize. So what are the specific provisions in 187 for adaptation and learning and observation over longer periods of time?
That I would consider, in a scientific way, a very weedy question. Guidance 187 is a lot of detail in it, but it is fairly broad. That's probably a question, to be honest, is best addressed to our experts back home. If you want to see me at the break, I'd be happy to take that question back, but I don't want to give you an inadequate response, and I think our folks back home will be able to give you the best response.
I will say broadly that if you look at the coordinated framework in the 1992 update, there is a principle that's articulated in those, and it was included in the slide that I presented at the beginning, talking about encouraging the agencies to continue to update their oversight of the products of biotechnology as their knowledge, as the science evolves in this space. So that is certainly an underlying principle that exists here. And how it relates to a specific product area, I think that's best. You talk to the agency about that. We have time for one more on this. Otherwise, we will move on to our next case study. One more over there. Okay.
Hi, Bignesh Ganapathy with the EveryLife Foundation. I'm curious about how the process at CVM informs the new drug application process at CEDAR, and whether any of the determinations need to be included in the application.
Good question. In some instances, there may be, for example, in this case study, there may be information about the milk that is relevant to both CEDAR and CVM. So there are mechanisms for the agency, or the centers rather, to communicate in some cases the data that suit one purpose may suit another as well. So, but again, I mean, that would all depend upon the specific product that you have.
And typically, the developer would be speaking with both of those centers in this case. And when there are areas where where information needs to be shared, then that would be done, with all three of those parties at the table.
Right, and I think one final point on this particular case study, if you are used to doing, producing new drugs and going with an IND, it's useful to know that with a genetically engineered animal, you should be going to see CVM earlier in the process than you normally would for an IND, because you've got investigational new animal drug as well. And so again, once that lineage has been established, you should go see CVM. So do not wait to go to see CVM once you're ready to file an IND. Go well before that.
Okay, thank you for those great questions. The final case study is a microbial product for industrial application. This is case study number 7 in the handout that you have in front of you. This is a hypothetical genetically engineered algae for biofuels. A unicellular algae is genetically engineered with a plant pest component to produce industrial oils for conversion into biofuels. The product is the eukaryotic microalgae Chlamydomonas reinhardti. It's genetically engineered to produce an enzyme that increases lipid biosynthesis. The extracted oils are later converted into biodiesel. The enzyme that increases lipid production was originally isolated from the soybean, glycine max. The soybean gene is controlled by the cauliflower mosaic virus-derived 35S promoter. The plasmid encoding the enzyme promoter and selection marker is introduced into the algae through electroporation. C. reinhardti will be cultivated in an open pond system. The remnants of the microalgae are intended for use as fish food. Which agencies have oversight and why? USDA has oversight because the microalgae are engineered with a plant pest component, the cauliflower mosaic virus 35S promoter. EPA has oversight because the microalgae are engineered for industrial use with genes from outside the genus Chlamydomonas, and as such, they fall under rules implementing the Toxic Substances Control Act. And the FDA will have oversight because the microalgae will be used for animal food. Okay, Mark.
Oh, okay. I'll go first.
Oh, are you not first, Mark?
I'm not sure. Originally it was Neil. But
Okay, Neil, go ahead.
Okay. So the USDA role would be very similar to what I've described in the other cases. I just mentioned that this case, this is GE algae in an open pond situation we've never regulated before. So the main difference here is that at the time of a petition for non-regulated, at the time of a environmental release, we may find that there's an exception to this confined field trial categorical exclusion, and the exception we make is for new species or novel modifications that raise new issues. Since we have never regulated an algae in this situation, there's a good possibility that we would decide to do an environmental assessment at the time of the field trial. And otherwise, this is very similar to what we've done in the other cases.
Okay, so as Robbie said, this is a microbe that's used for a purpose that's not excluded by TSCA. It's considered new because its genes come from more than one genus, which we call intergeneric. So if this microorganism is used in a contained structure, and it complies with the list of best practices that occur in our regulation, no reporting to EPA at that stage is required. Structure is interpreted broadly by EPA. We're not just talking about a lab bench in a building. For algae, this is important because photobioreactors could be included as contained structures. Of course, that means that all the piping and all the ancillary material associated with a photobioreactor have to be truly contained. So if there is any question about the design of the structure, as Jason has been saying all along, we agree, please contact us, talk to us, and we can go through some of the details to be sure that there is no need to provide a formal report to us. So EPA does not separate small scale from large scale in determining whether there is a need for oversight. A release is a release, and thus a TSCA experimental release application, a TERA, would be needed as long as there is an intended release. This is an open pond, intended for an open pond system. Presumably somebody will be testing this in an open pond. And Robbie, could you go back to the image? And let's see if I can borrow this. Because I want to show you a little bit about scale. Back here are some mini ponds that are basically something like 1,000 liters. Then it can go up to something that's about twice the size, and then something that's quite a bit larger, all of which are found in one particular facility, in this case a test bed in Arizona, which might be a kind of place where an experimental release could take place. Beyond these sizes, somebody may want to then go 5, 10 times as large out in the middle of the desert someplace to do their experimentation to make sure that their organism works on something approaching a commercial scale. All of these could be wrapped up into one single TERA. But if anything goes wrong or changes or something shows up in the course of doing experimentation as one goes up in scale, it might not have been covered by the data supplied to us, all those kinds of data I described earlier. It might not have been described well enough to support an acceptance of going forward with that particular TSCA experimental release application for all the scales. So what we might expect to see in a case like this would be a progression, a succession of individual TERAs over the course of different growing seasons and different scales that are appropriate for this particular microorganism. Assuming everything goes well with this experimentation, the data that are gathered from the experimentation are going to be useful later on for evaluation and commercialization, then presume that submitter would want to make some money off of this, and they would have to then come to us for a microbial commercial, an MCAN, microbial commercial application, notification. So in the case of the TERA, we have to formally give an approval before they can go forward. In the case of an MCAN, we do a review regardless. If something is new, it triggers oversight. And so we would go through a review. If at the end of the review we find that there are no issues, that we cannot make a finding that there is unreasonable risk to man or the environment, all we have to do is stay silent, and after 90 days, The submitter is permitted to go through with whatever commercial activities they choose to do. Now, let's say this submitter said, we want to make this organism at one site. We want to have one particular use, making biodiesel, and that's it. We have to consider that somebody else may choose to produce the same organism somewhere else or for a different purpose. So we have to think of all the plausible uses for the organism, all the plausible production methodologies, and consider risks overall. We may find that we're uncomfortable with saying nothing, and that we believe that there should be some limitations on the production from a risk basis, or production or use or whatever, commercial application. We can and would then consider working with the submitter to make sure that there are appropriate limitations for that production or use of the microorganism, that a consent order would be drawn up, and the submitter would agree to these conditions, and therefore limitations would be placed on the commercialization of this microorganism. It's entirely possible that additional data could reveal that additional uses or additional production at different sites might be appropriate, and the submitter might choose through maybe additional terrorists to gather that information, provide it to us, and provide us with reasons so that we could lift some of those limitations or all of the limitations. That is part of our process. It's also possible that we may find that we have to unilaterally impose limitations. We prefer that we don't do this. In this case, we're only increasing the production of a natural
Mark, I'm just going to try to get you to wrap up here so we can do them. Okay, good.
Okay. So if there is no new chemical being produced, then there will not be a need for additional oversight. If, in fact, they were making this organism produce a chemical it didn't naturally produce, and that was not on our inventory of chemical substances, a separate review of the chemical would be needed. A pre-manufacturing notification would have to be supplied.
Jason.
Sure, and if the spent algae were to be used for animal feed, again, it would have to be safe and lawful for its use as animal feed. One of the considerations would be whether, for instance, the algae had aspects to it such as remnants of the fermentation material or the substances produced during fermentation or substances added to the fermentation to control it, whether those substances present in the spent algae would be important to consider in terms of safety for animal feed. And all those factors would be considered in whether something like this would require a food additive petition or whether it would not. And so this was a great example of the desire or the need to consult the agency early in development. And in particular, for things like this, it's useful to think about your product life cycle very early in research and development so that you are consulting with the agency before you have a lot of spent algae that you are looking to get rid of in some way. So again, think ahead and come and talk to the agency about things like this, because this is where we can be very helpful. Perhaps it's something as simple as a tweak to fermentation conditions may mean a regulatory hurdle versus something that is much less of a hurdle. So, thank you.
Great. So let's do questions on this case study. Right up here at the front. Why don't you just go ahead and we'll start with you and I can repeat it.
Okay, sure. My name is Alan, and I have a question. Is there a difference if you have the algae express the oils transiently versus having completely changed transgenically? Is it for regulatory purposes? So it would be like a on and off part.
So Mark, why don't you start
I was
with
gonna
that?
say
So
that-
the question is whether if the product being produced is expressed
Yes.
so if the expression levels vary over time.
So that would be part of our consideration during the course of our risk assessment, whether that transient production would have an effect on the environment, for example, and it's just a specific part of the assessment would be case specific, and we would just have to evaluate that question in detail. I can't give you a yes or no answer right now.
Anybody else? Jason?
Yeah, I think Mark's point is very good. I mean, for us, usually it would depend upon what in fact is going into the food or the feed and what is its state at that point and what comes along with it as residual material. And, you know, I think, again, it's a good question to come and talk to us about because the specifics there may be important.
Yep. Thank you. Yes?
Hi, thank you. Another question.
Name, please.
Dana Pearls.
Okay.
So I have a question about containment. Earlier you addressed the difference between confinement and containment. And in this case, and in the case of a lot of algal biofuels genetically engineered using transgenics or newer gene editing techniques, it's questionable whether or not containment is ever 100% effective. And so you mentioned that containment, that you try to ensure that the probability of escape is close to zero. But in the case of both open tanks and also containment that might just be plastic bags or other forms, what is the process to ensure that there is no escape, and in the case that even minimal escape, even if it is close to zero, that may have significant environmental impacts? How do you address those?
So I think it's important to note that the discussion about containment and confinement at the beginning was from USDA, and that was a discussion about that was not specific to the algae. And then there was, EPA also has confinement.
Okay, so specific to this example.
Let's Mark.
Yeah. So no, we don't use the term confinement.
Right.
And
EPA does not.
No, but you are asking a very good question, which would be the kind of question we would be asking when somebody would be consulting with us to inquire whether or not their particular system is truly contained. We would expect to see some kind of data or information that would establish whether or not something is completely contained. We're looking for secondary containment in case there are leaks, things like berms and collection of material of fluids or something like that that might come out, or use in a desert environment where escape is not in the immediate area of the production can be controlled, if in fact that's an approach that somebody might want to take. So yes, that's an interesting question, but it is something we would evaluate if somebody would come to us beforehand.
And Neil, would USDA have be addressing those in this specific case study?
It's possible. We have permitted some field trials of algae that was grown, like you were saying. We sent someone out to the site and looked at it. It was basically in a desert environment, and we felt that if there was a breach of that contained bioreactor, that the algae could not grow in that environment. So that's how we considered it.
And so the regulations for what's considered to be closed containment and open containment, given that containment isn't in fact 100% effective, do the regulations apply similarly, whether closed or open?
So is that question for USDA or for EPA?
Both, thank you.
Well, in this particular case, we did permit it, so we were considering it.
Yeah, not in this case, but in the one that you were...
I'm sorry, the one that
the
you
previous
referred
product.
to of a bioreactor in an outdoor environment. We did consider that confined, and we did permit it. we generally have used, where we have not issued permits, have generally been in greenhouse situations. And yes, I suppose you could have some pollen escape from a greenhouse. I mean, it requires...
But for algae.
But for algae, we would not regulate algae. For example, if it was a laboratory and it was just bioreactors in a four-wall environment like this.
So Mark, can you address how you think about containment, maybe in a little more detail?
Yeah, I'm not sure I can do it in a lot more detail because a lot of it is going to be depending upon the specifics. We will have our engineers look at the production system. If there's any question about whether something is contained, and we have had cases already where people have come to us. We do have some experience with algae and with both in both contained systems and open systems. So we have we will have people developing ways to look at containment. Right now, our points to consider document, and this is something I haven't had a chance to talk about yet, but we're in the process of revising our points to consider document to include some specifics on dealing with algal production. We're doing that right now, so I can't give you any details, but it's a certain, we had a public meeting on this issue back in September, and we're dealing with some of those comments and issues at this time. I guess I really can't answer any more than that.
Great, thank you. We have time for one more question. Is it okay if we let somebody who hasn't asked yet?
Hi, this is Vince Seawald with DuPont. Question for Jason. You were kind of light on FDA requirements for the algae. If they end up in animal feed as a feed ingredient, it is likely that they would be considered a food additive. So my experience is one would get in touch with CVM very early in the process to verify that, and then establish a plan for either a GRAS determination as an exemption to food additive. So could you elaborate on that option for GRAS determination?
Well, the most I can really say is, again, it may depend upon the specifics associated with the product and what our scientists at CVM come out. But certainly, you're right. There is always the possibility that something could be generally recognized as safe, and you could voluntarily submit a notice indicating that your product is generally recognized as safe. But I think you made what I think is the excellent point, think about this early in development, because the regulatory process may be important to your regulatory and development timelines. And so I think really what I heard from your question is primarily, go talk to the agencies early in the process. Whether or not they would consider this particular substance a food additive or a substance that is generally recognized as safe may hinge upon the specifics of the situation. For example, some oils may be harmful to certain animals, while others it may not be. And so it's very hard to make that specific case without knowing, down to the finest detail.
And I would like to say amen to what Jason said about coming to us early. This would go to the dealing with, for example, the question that was just asked about how do I know whether or not my system is contained. Come to us first. Ask us first before you start developing a system or get very far along with it.
Okay, so with that, we are finished with the case studies. We will have a little bit of an adjustment to the agenda here in order to make sure that we accomplish everything. Do you want to give an update on that, Mike?
So the bad news is we're about 25 minutes behind. The good news is we dropped a 25-minute session. So we're right about where we need to be. The break will start now, It will run 11:30 to 11:50. When you come back at 11:50 for the breakout session, they will last the full hour as in the agenda, and then we should be able to start the public comment period as scheduled at 12:50. So the locations for the different breakout groups, the governance group will be in this room. I would say on this side would be better because there's a monitor on the floor over here. The communications breakout will be in conference room A, which is just on the other side of the registration tables. And the regulatory certainty will be in conference room B, which is the one closest to the registration tables. The hotel right over here has some salads that they sell, and there's a food truck out this way, and there's vending machines in various places if you'd like to get something to eat in the next 20 minutes or so.
And then do you want to talk about how we're going to do the comments? We'll
probably repeat this, but the comments will be done in alphabetical order, and we will have the listing in the room at the beginning of that session and during the session, so you can see when your comment will come up.